This is part 2 to chapter 10 videocast lecture on sexual reproduction and genetics. In this section of the video lecture, we will be focusing in on Gregor Mendel's work and taking a look at how to solve Punnett square problems. Gregor Mendel is often called the father of modern genetics. He was an Austrian monk, so he lived in a monastery, and one of his jobs at the monastery was to run the garden. And in observing the life that was growing in the garden, Mendel, kind of having a little more math and science background than your average Joe in 1866, started to notice some patterns of how different plants grew with different types of flowers or different types of seeds. And he wanted to know why. So he was really the first guy to document his experiments and observations about why things look the way that they do. And his most famous study was using pea plants. So he was growing garden variety peas. They are perfect to use for genetics experiments because, first of all, they grow very fast. The traits are very easily seen. Plants were either tall or short. They either had purple flowers or white flowers. And you can tell a pea plant to mate, essentially, with another pea plant. So you can manipulate fertilization. You can self-fertilize pea plants. You can also cross-fertilize them. And the pea plant isn't going to say one thing to the experimenter one way or the other because they're a plant. These were the different traits that Mendel took a look at, and he had literally thousands of plants that he was keeping track of for all of these different characters. He looked at seed shape and color, the flower color, the pod shape and color, looking at the flower position on the pea plant when it bloomed, and how tall the plant was. So what he did, in order to start with a clean slate, if you will, he self-fertilized many generations of plants. So he would get and end up with what he considered a true breeding or a purebred plant. And he marked those with P for the parental generation. So in this example here, he took yellow peas and decided to cross-fertilize those with a plant that produced green peas. So he cross-fertilized the pea generation, ending up with what's called the first filial generation. Filial is Latin for son. And so these were like the sons of the pea generation cross. Now what happened when he took all of the yellow peas, cross-fertilized them with the green peas, the F1 generation, the first generation that appeared, had all yellow peas. It was as if that green pea trait just disappeared from the population. Now this made Mendel question what he was doing. At the time, a very common idea was if you took a yellow pea and a green pea and you mix them together, you'd get a greenish yellowish pea. But what ended up happening was those green traits completely disappeared. Then he decided to take those plants of the first generation, that F1 generation, and he self-fertilized those. And in the F2 generation, so this would be like the grandsons of the pea generation, then all of a sudden those green peas came back, but they didn't come back in an even ratio. There was always this ratio of 3 to 1, no matter how many peas he counted from these plants. There was always one character that was always 3 times more dominant than the other. So this is the whole thing from start to finish. We have the pea generation. We cross those, all yellow peas in the F1 generation. After self-fertilizing those, that green trait came back, but at a much reduced rate than what Mendel thought. So this was for every trait that he tried, whether it was height or flower position or pod shape or pod color. It was always this 3-to-1 ratio in that F2 generation. So after working with this for a while, he figured something had to control what we see. And he called these things factors. However, we know now that they're actually called genes, and these factors or genes come in pairs. So you get one pair from the Ma organism and one pair from the Pa organism. So you get one trait from each parent. So these factors, or what we call genes, come in pairs. And every gene has a form. We call the forms of traits alleles. So for example, if we were talking about alleles for eye color, we would have blue eyes, brown eyes, green eyes, hazel eyes. Those are all of the different forms. So when Mendel was doing his experiments, he realized that one of these factors, or alleles, were dominant. They would mask or dominate the expression of the other allele. So we represent dominant alleles with capital letters. So we know that it's dominant. So for the color, since that was that 3 in the 3-to-1 ratio, there was more yellow than green peas, we have a capital Y for yellow. Recessive alleles, the green peas in the 3-to-1 ratio, those seem to get masked or hidden in the presence of a dominant allele. So we represent recessive alleles using lowercase letters. So in the color example, if capital Y is yellow and yellow is dominant, lowercase y is recessive and that will stand for green. Now, I know what a lot of you are saying, why can't I use Y for yellow and G for green? Because that's the way it is. If you use 2 different letters, that means you're suggesting 2 different alleles or 2 different forms of a trait. Allele pairs must have the same pair of letters. If you wanted to use the letter F, capital F is the dominant trait, lowercase f is the recessive trait. That's the way it is. It's not going to change, so when you do your practices, you have to get used to using the same pair of letters to represent one form. So what Mendel realized was that the F1 peas were the same shade as the P1 peas, but different alleles made up that F1 generation, different forms. So here we can see with corn, believe it or not, purple is actually a dominant color for ears of corn, and yellow is recessive. Don't always equate dominant with more popular and recessive with less popular. That doesn't have anything to do with popularity or how much it's present in a population, because recessive traits can actually be more popular in a population. It all depends on what alleles the parents have. So here we have purple and yellow ears of corn. We cross those because purple is dominant. All of the ears of corn have purple kernels. However, because that yellow parent contributed 1 recessive allele, the F1 generation corns, even though they look like the purple of the P1 generation, don't have the same alleles as the original purebred parent. If both alleles are the same, meaning the same level of dominance or recessiveness, then we call those homozygous alleles. So homozygous, you will have capital, capital, or lowercase, lowercase, because remember, homo means the same. Heterozygous alleles are different. You will have 1 dominant and 1 recessive allele. So you will have 1 capital letter and 1 lowercase letter. And just so you note, the dominant allele is placed first. So even though Mendel knew nothing about chromosomes, he knew nothing about mitosis or meiosis, He was doing all of these experiments based on his visual observations. So even though he didn't know it, he was actually working with the genotype of these pea plants. The genotype is the actual genetic makeup of an organism. It's the allele pairs that the organism has. So for the genotype of capital Y, lowercase y, capital Y came from one parent, lowercase y came from the other parent. So the genotype essentially is what you are. Now we also have what you look like. The phenotype is the observable physical outward expression of the characteristics. That's what you can see. So really Mendel, even though he was using and collecting his data on phenotypes, he learned that those observable characteristics came from those factors or genes from the organism. So the genotype of capital Y, lowercase y, would be and look as a yellow pea plant.